You're listening to Medscape's In-Discussion series on respiratory syncytial virus in adults, a podcast where thought leaders and clinical experts share their diverse insights and practical ideas for optimizing patient care. This series is hosted by Dr. Forest Arnold. Dr. Arnold is the chief of the University of Louisville Division of Infectious Diseases and director of the Center of Excellence for Research in Infectious Diseases. He also acts as the co-editor-in-chief for the Journal of Respiratory Infections and program director for the Clinical Fellowship in Infectious Diseases. Relevant disclosures can be found with the episode show notes on Medscape.com or the Medscape app. The topics and discussions are planned, produced, and reviewed independently of advertisers. This podcast is intended only for U.S. healthcare professionals.
Hello, I'm Dr. Forest Arnold, and welcome to Medscape's In-Discussion series on RSV in adults. Today we'll be discussing RSV vaccines with Dr. Barney Graham. Dr. Graham is a senior advisor for global health equity and professor of medicine and microbiology, biochemistry, and immunology at Morehouse School of Medicine. Welcome to In Discussion.
Thank you. I'm glad to be here. Thank you for the invitation.
We can't talk about the new RSV vaccines without talking about the beginning of the vaccines, which wasn't 5 years ago. It was 1965, just a decade after the very successful polio vaccine was released. Could you take a couple minutes and review those first years and the children's studies?
Yes. RSV was discovered in 1956, right as the polio vaccine was having so much success. And the initial attempts at making a vaccine were, at that time, to isolate virus, grow it to high titer, and inactivate it, and that would be the vaccine. And that was done with respiratory syncytial virus, and a whole inactivated, formalin inactivated alum adjuvanted vaccine was tested. And in 1965, there were studies done in 4 cohorts of children, and in the youngest age cohort, where vaccinations were started before 7 months of age, in children who had not yet experienced an RSV infection, there were some problems. And in that cohort of young antigen-naive children, out of the 31 vaccinated, 20 became infected during that winter season, just after Christmas of 66-67 season. And 20 of them had to be hospitalized, and 16 of the 20 infected, 16 had to be hospitalized. 2 of those 16 children died. So that was a dramatic setback for vaccines in general, but it was a real surprise and put a halt to vaccine development for RSV for several decades until the immunology and pathogenesis were better understood.
Right. So in the mid-1980s, the fusion glycoprotein F, which we'll call F protein, was isolated. And what is the importance of that F protein?
Right. Well, in the 1980s, you started getting sequences for RSV, monoclonal antibodies, and isolation and characterization of the proteins of the virus. And there's 3 surface proteins, the SH, small hydrophobic, G, and the F glycoprotein. The F is considered to be the most important for vaccines, and antibodies targeting F can neutralize the virus or prevent virus infection. And F is important because it is the protein that mediates membrane fusion. It helps fuse the viral membrane to the host cell or target membrane. And that's how the viral genome gets into the cell to start the replication process. So if you can prevent that F protein from doing its job, then you can largely prevent infection of the next cell.
Okay, and then there is a pre-fusion form and a post-fusion form. And what is the significance of those 2 forms? And what are they fusing to?
Right. So the F protein is referred to as a class 1 fusion protein. It's a type of protein on envelope viruses like parainfluenza, HIV, Ebola, viruses like that, including coronaviruses. And on RSV, the F protein is anchored in the membrane. And it starts out in one conformation, like a spring-loaded trap. And as it approaches the cell, it unfolds. The top of it unfolds, and it inserts itself into the target cell membrane. In this case, for RSV, it's mostly the ciliated bronchial or epithelial cells and the type 1 pneumocytes in the alveolar space. And that attachment then leads to a rearrangement of the protein where the 3 identical parts, it's a trimeric protein, the 3 identical protomers grab the host cell, and then the 2 ends, the one end inserted into the host and the end inserted into the viral membrane, come back together. They're pulled together. And that creates a fusion pore that allows the virus replication process to start. And so moving from pre-fusion to post-fusion, you're also moving from functional to non-functional form of the protein.
And it's the post-fusion that is short-lived, I guess. It's usually in a pre-fusion state. Is that correct?
On the native virus that's budding off of cells, the protein starts as a pre-fusion. So that is the spring-loaded form. The post-fusion is a very stable molecule. And before work in around between 2010 and 2012, we didn't really understand the important differences between these two forms of the protein in terms of what antibodies recognize. But the post-fusion form of F is very stable. Once it makes that rearrangement, it is an extremely stable molecule. And so it's relatively easy to make. It's easy to store. It's easy from a manufacturing process. And it is what you get when you isolate the protein from a virus or make the protein in cell culture. So the post-fusion is what people had to work with up until around 2012 or 13.
Okay, and then new vaccines utilize that F protein as part of their mechanism of action, correct?
That's correct. When vaccine studies resumed in the 1990s, after there was a better understanding of the vaccine-enhanced illness, which only antigen-naive people are susceptible to that kind of event. Once people are infected with RSV, which starts early in life and then repeated throughout life, once people are primed in the right way for an immune response, vaccination or infection doesn't result in this kind of vaccine-enhanced disease. And so in the 1990s, immunization studies were resumed, and there were 5 phase 3 studies with what turns out to be the post-fusion F molecule, and they all worked to a certain extent, but they didn't boost neutralizing activity very much, only maybe 2 to 3-fold on average. And so none of those phase 3 trials were able to progress. They weren't considered, they didn't meet their primary objectives. The big thing that happened in 2012 and then published in 2013 is that we were able to capture the atomic level structure of the pre-fusion form of the F protein. And once we had that, and that was done at the Vaccine Research Center at NIH, in collaboration with a fellow, Jason McClellan, who was in Peter Kwong's lab and my lab, we worked on this protein, got the structure, were able to stabilize the structure by putting in some internal mutations and holding it together with what's called a trimerization domain on one end and putting in disulfide and cavity filling mutations inside. It holds it in the right shape and doesn't let it flip around into the post-fusion form. Once we had the protein in that form, it turned out to be a much better vaccine antigen. So now in animals, for instance, you could you could elicit 40 to 50, sometimes 80-fold higher neutralizing activity from the pre-fusion molecule than you could from a post-fusion molecule.
Okay, and we'll get back to those neutralizing antibodies. But first, we have to sidestep and talk about COVID-19 for a second. There's 2 vaccines out that just came out now, and other drug companies are probably going to follow suit with new FDA-approved RSV vaccines, which makes it sound like the technology has happened after COVID-19 vaccines because we're after the COVID-19 vaccine. But COVID-19, in a sense, got in the way of making the RSV vaccines and actually provided the technology and lessons learned to create the COVID-19 vaccine so quickly. Is that giving too much credit to the RSV vaccine?
Well, no, I don't think it is. The RSV vaccines, from that initial discovery of the pre-fusion conformation and that being a better vaccine antigen, RSV vaccines have been on more of a traditional development timeline. And so our phase 1 trial that we did and published in 2019, it took that long to evolve. And as the companies have developed their products based on that confirmation, it's gone through about a 10-year development timeline, which historically is actually relatively fast for vaccine development that has usually been measured in decades. And this is only 1 decade. But the concept of holding this fusion protein in its pre-fusion conformation is what led Jason McClellan and I. Jason was starting his own lab at Dartmouth Medical Center, and we decided to extend the RSV findings to another envelope virus, the coronaviruses. And so working on MERS and then the first SARS and eventually HKU1, which is one of our endemic coronaviruses, we were finally able, with Andrew Ward's collaboration at Scripps, to get the pre-fusion form of the spike protein of a human beta coronavirus. And that allowed us to find stabilizing mutations. And that actually was used in prototype vaccines that we knew by 2019 could protect animals from lethal challenge with the MERS coronavirus. So the RSV, this concept of the pre-fusion stabilized protein being a better vaccine antigen, came from RSV, was extended to coronavirus, and is what prepared us in the field for rapidly making coronavirus vaccines in 2020.
Okay, so let me summarize to this point. In the mid-1960s, we had a very revealing but horrendous study where children who were vaccinated for RSV, many were either hospitalized and 2 died. And then 20 years later was the isolation of the glycoprotein F, but it really wasn't until it was outlined in an atomic form that we made progress. And then you move forward some more, and they're working on, you guys, RSV vaccines, and here comes COVID-19. And so the technology gained from RSV allowed a rapidly ability to form a COVID-19 vaccine. Now we're 3 years out from COVID-19 vaccine, and we've got our first RSV vaccines. Now, these are indicated for older adults. And my question now is, why aren't the RSV vaccines that are out now also for children?
Right. So the field spent 20 to 30 years working out the safety, and then it worked another 20 years or so working out a way to make vaccines effective. And these new vaccines, based on proteins, subunit protein, they are being used initially to boost adults who have already been primed with prior infection. So the vaccine for adults, which we know can be given safely because everyone's already been infected with RSV before, are being used to boost immunity in adults. And they can boost immunity to supranormal levels, to levels that are found in very, very few people from natural infection. And so they can provide a level of protection, usually for severe disease up above 90%, and for mild disease even above 70%. So these vaccines are effective in the elderly. They're also effective in boosting women of childbearing age or pregnant women enough that their antibody can be transferred to babies and protect the babies for around 6 months. And so we do have an approach that will be available for babies, but it comes through the mother or through antibodies made in a factory and given directly to babies at birth that can protect them for around 6 months as well. The thing we still don't have, and the thing that is still being worked on, that will probably take a format other than a purified protein, is a vaccine for the 6-month to the 5-year-old child. And most hospitalization, most lung damage happens in children less than 6 months of age, but there's still a lot of disease and hospitalization in children between 6 months and 5 years of age that we would like to eventually have something for, and that is still something that's being worked on.
Okay. Have you described disease enhancement? And by that, I mean RSV vaccine-associated enhanced respiratory disease. Is that a problem that children or, infants experience that adults don't because they've already been exposed to RSV?
Yes, the vaccine-enhanced illness associated with RSV and immunization in infants, what's called vaccine-associated enhanced respiratory disease, or VAIRD. That is a syndrome that has an immunological basis. And there was found, over the years, to be two problems. One was an antibody problem, meaning that there was a lot of antibodies induced that could bind the virus protein, but did not neutralize the virus infection. And so because the virus could still grow, and because the antibody levels were high, it was found in those early studies to have caused an immune complex mediated disease, where you could find immune complexes in the small airways that activated complement. And we think that is part of what happened originally. So part of the problem to fix that vaccine-enhanced disease is to keep the protein and the conformation that will elicit more potent, effective antibodies. But there was also a T-cell problem, and it was found in animal models with some corroboration in human studies that in people who are primed with a protein or whole inactivated virus type vaccine, that when they are challenged or infected with RSV, they get more of an allergic inflammation. They get what's called a Th2 response, or a CD4 T-cell response that includes IL-4 and IL-5 and IL-13, which means you get a lot more mucus, you get a lot more bronchoconstriction, you get a lot of eosinophils and neutrophils. And that kind of response is not as effective at clearing RSV, and it causes a lot of problems with airway obstruction. So that kind of response, that kind of immune response, is something that needs to be avoided, especially in the very young, small child. So making sure that we have the vaccines set just right and all the children primed in just the right way to create the best possible immune response is going to take a little longer than it has for the adults, because in the adults, the T-cell response is already set, and all you have to do is make sure you're boosting the right antibodies, which we can now do with this prefusion F protein.
Okay, so the immune-mediated response in children is what causes them a problem if they receive the same kind of vaccine that we're giving to older adults. Is that immune-mediated response the problem with the whole inactivated vaccine back in the 60s?
Well, what we know is that the way they made that vaccine in the 60s was to heat it at 36 degrees for 72 hours. And we know that if you do that with virus, that all of the F protein flips into the post-fusion form within about 24 to 48 hours. So that vaccine that was given back in the 1960s was a post-fusion F antigen. It didn't have any pre-fusion F on it. So it's possible that the pre-fusion F protein by itself could be a solution for young children. And that if we have the antibody right, the T-cell problem won't be an issue. But because there's a little uncertainty about that, there's going to have to be a lot more work. And most of the vaccines being developed for the young child are live attenuated viruses, which would mimic better a natural infection, or potentially gene-based delivery of vaccines that would mimic infection more and also elicit what's called a Th1 type of CD4 T-cell response that would have more interferon gamma and less IL-4. So those kinds of vaccines are coming along, and it's possible that just the protein that's in the right shape would work, but because of caution and because most vaccine developers are mostly focused on safety first and efficacy second, that, this is going to be done with all caution.
I see. Today we've had Dr. Graham discussing several important parts of the RSV vaccine. We've talked about its beginning in the 1950s. We've talked about the importance of the F protein, especially the pre-fusion form. We've talked about its impact on the COVID-19 vaccine and how it made that possible quickly. And we've talked about disease enhancement as well as neutralizing antibodies that would be present in mothers who would be vaccinated, which may be available soon, and the protection it could offer fetuses in the first 6 months of life. Thank you so much for joining us. This is Dr. Forest Arnold for In Discussion.
Thanks for listening to Medscape's In Discussion Respiratory Syncytial Virus in Adults podcast series with our host, Dr. Forest Arnold. Be sure to look for more In Discussion episodes wherever you get your podcasts. Check out Medscape.com or the Medscape app for show notes, links, and more information on respiratory syncytial virus in adults.